So let's see inside the cell. Where does this happen? Where does this RNA processing and mRNA translation is happening? First, we have to look at the structure of RNA first. So from DNA, there are promoter region for transcription. And in fact, not all segment of DNA is actually a coding region. So there are regions which is not involved in direct coding that we call as like intron, because it's inserted probably. And the rest part is called exon, which is a coding region. So once we transcribe, then what we get the original RNA looks like this: exon number 1, intron, exon number 2, intron, like this. And so intron is in fact not necessary for translation. So we actually take this on apart and then join these exons together. So joining is called splicing. So we call it as a splicing. And this part is part of RNA processing. So this contains the coding information. So we call it a messenger RNA transcript. And after translation, we can create a polypeptide chain or protein. So these coding regions of DNA we call exons. And while non-coding regions, we call it introns. So primary RNA transcripts undergoes splicing to remove unnecessary introns. And let's see how this happens inside a cell. So this is cell double membrane, and this is a nucleus, and inside there's a chromosome, and inside DNA. And DNA first make RNA transcript. Transcription makes an mRNA, and that mature mRNA will come out of this nucleus and transport it to cytoplasm. And inside the cytoplasm, there exists a ribosome. And this single strand messenger RNA transcript is now ready for producing polypeptide chain by translation. And that scientists consider, how does this happen? We do need some adapter molecule which corresponds these 3-letter information into one single amino acid. So there comes an idea of a transfer RNA. And that is happening at this complex machinery of ribosome. So this nucleus containing eukaryotic gene, there is a promoter region containing binding site for general transcription enzymes and other transcription factors. And the introns are spliced and removed to yield an mRNA transcript, which consists only exons. And the resultant mRNA is then translated into a protein. Oh yeah, so this translation occurs at the ribosome. So now thinking about the genetic code, how to convert a linear sequence of nucleotide in mRNA, which is composed of A, U, C, G of 4 types, into a linear sequence of amino acid, which is building blocks of a protein. And we know there's about 20 amino acids. So that is having this mRNA transcript, which also has a 5' end to the 3' end. And interestingly, now let's take it, because we will discuss more later. So this 3-letter of nucleotide corresponds to 1 amino acid. Then this transfer RNA should be adapting this. So one side, such as, let's say, there's a GUA, is supposed to produce a valine. Then there's a complementary sequence C, A, U. And one side is, and the other side must have this valine amino acid, and they will attach to this as a growing polypeptide chain. So this polypeptide synthesis is that amino acids are added to a growing polypeptide chain by tRNA. This tRNA serves as an adapter molecule, and each tRNA should contain the opposite of this codon, so we call it as a anticodon, so which is complementary 3 letters on one end, and the other side, the corresponding amino acids should be attached. So 3 mRNA bases we call triplet or codon. This is read at one time by an incoming tRNA. So if the anticodon of tRNA is complementary to the codon region in the mRNA, then amino acid from that tRNA is added to this polypeptide chain. So now you will have a question like, hey, why is the codon made up of 3 amino acids? In fact, in history, figuring out this actually led to Nobel Prize again. So this is called finding a genetic code. And you see from this, based on the mRNA, so there's 4 types of nucleotide: uracil, cytosine, adenine, and guanine. And so the first part is this, and the second letter can be also UCAG, and the third letter is UCAG. Then how many of these 3-letter sequence can create, which is 4 times 4 times 4, which is 4 to the 3 or 64. And what is the final number of amino acids, which is 20. So, you know, the genetic code is actually not like exact 1-to-1, but any 20 amino acid can come from maybe several different mRNA sequences. And this finding and this genetic code, we call it then nature as a degenerate, only used in biology as the term, the ability of elements that are structurally different to perform the same function or yielding the same output, called degenerate. So let's think a little further about the genetic code. How many nucleotides of mRNA are required to encode the 20 amino acids? And we said 3 bases along the mRNA chain for 1 amino acid is called codon. That's a minimum unit of genetic code. And interestingly, there are codons which is leading as a start codon and stop codon. So you can see here, like these 2 UUC will code for phenylalanine, and these will code leucine, and AUG will encode methionine. And you can see there's like stop codon, stop codon, stop codon. And this methionine AUG is in fact start codon. So what it means is the translation event starts at this start codon. So it always starts from AUG on mRNA, and it terminates when it sees the stop codon. And interestingly, there's a 1 to 3 stop codon. First is UAG and UGA, UAA. And so if I want to summarize the transcription and translation by comparison, so transcription starts on DNA segment, which is called promoter region, and then it stops at the terminator region. While translation is based on mRNA, and where it's AUG or called start codon, and the other UAA, UAG, UGA as a stop codon, which means translation stops there. And the degeneracy, as I alluded, any 20 amino acid can come from several different mRNA sequences. I'm going to discuss this one further.